So, to begin, we'll talk about plant hormones. On the picture here, you see the chemical formula of abscisic acid, which is an important plant hormone. And as you see, these molecules are all organic, so they have a lot of carbon atoms and um, also a lot of hydrogens. Here you see a CO double bond group, a carbonyl group. Here are methyl groups, an alcohol group. So this is quite an interesting molecule from a chemical perspective, also with lots of double bonds that can also contribute to color of that molecule. So, as I already said, we'll be talking about how plants react to stimuli. For example, light. They have to know when it's day and when it's night, even if they don't really have a sleep or activeness and sleep cycle, but they want to grow towards light. And here, this is called like a way to react to the stimuli is through tropism. This is growth towards a stimuli. For example, in phototropism, it is towards the sunlight or towards light. And it's actually the case that plants that are next to street lamps are like get like leaves earlier in the spring than plants that live in shadow like uh, regions with a lot of shadow and not a lot of sunlight or light from houses or street lights. And therefore, you see that light is an important stimuli that they can react to. There are also other forms of tropism, for example, gravitropism. Like gravitation is also something they have to react to. For example, in in gravitropism, the plant knows where the bottom is and where it goes up into the like this like out of the ground, and this is important. For example, if a seed is inside the soil, it has to know in which direction it has to grow. It would be like not a good outcome if it grew downwards, because then it would never reach light, would never be able um, to to make photosynthesis and create its own energy, and therefore it would die. And therefore, the like the good thing is it knows how to move upwards against gravity, and then it can reach like the end of soil and like the air. And salt and minerals in soil are also something they have to react to. And this is especially important as they have to know where the salts and minerals are that they take up through the roots. And only due to these things they can sustain their functions, as they are quite important for that. If they are damaged, they need to react to it. Like they also have like feelings is not the right word, but they know when a leaf is broken away and when harm is made to them, and they can do that, for example, through through biochemical reactions. For example, when a um, fungi infect a plant leaf, they might stop photosynthesis at this particular point, and then the fungi can't grow anymore because there is no nutrients that are produced, and so the leaf dies. But with it, the fungi, that's kind of the immune system of plants. Another thing that is important is temperature. I think with climate change and global warming, we have all heard that certain plants are more susceptible to high temperatures, temperature changes, and that they suffer of higher temperatures and maybe even die because of them. And they also get stressed when it is too hot. Moisture is also something they have to detect. For example, they need to grow their roots to places in the soil that are wet so they can sustain their water needs. There are other ways they can react to stimuli, for example, physiological changes. By, for example, closing stomata, they can reduce the amount of CO2 they take up or the amount of water they lose to the environment. Let's define the term hormone. As we saw on the first slide, it is a chemical molecule. It is a signaling molecule that can be transmitted throughout an organism. So it is produced in one organ and then transported to others. There the receptors are. So it travels through the whole plant, also through the whole body in humans. And then when it reaches tissue or cells with specific receptors, it can begin to, it starts to, it's like it can bind to it and then enter the cell, and there's some function being carried out due to it. Hormones also work in the smallest concentrations, meaning that you don't need a lot of hormones for a specific outcome. Only a small amount is enough. For the transportation of hormones in plants, there are two ways. One is the phloem and one is the xylem. The xylem are the vessels for the transportation of water and minerals from root to leaf. So from the root upwards in the shoot. And the phloem works a bit differently. It is basically like it transports materials like soluble glucose and other nutrients from where they are produced. For example, with glucose in the leaves, it is produced, but in other parts of the plant, for example, in the fruits, in the roots, they are needed, the nutrients, and so it is transferred from high concentration to low concentration. This is a core concept in biology that things move from regions of high concentration to low concentration through a process called osmosis. And the difference is that xylem are dead cells, so they are like wood. They aren't living anymore, whereas in phloem, the cells are still alive and transport the nutrients. There are other mechanisms that are a bit slower, for example, just from cell to cell through transport proteins within the cell membrane, but this is very slow and not suitable for hormones that should reach the region where they are needed fast. Here is an example of how hormones work. So there is a stimuli, and then the hormone binds to the receptor of the cell, and then it is transducted through relay molecules. And they make the signal stronger, for example, through second messengers, and then within the nucleus of the cell, they have some effect that could be changing of transcription factors and changed gene activity, and that can cause, like, for example, certain proteins to be produced that weren't produced before, or certain enzymes that can maybe digest something or speed up a chemical reaction that is necessary when a certain stimulus occurs. Here is an example. in potatoes, they like when they are in the dark and it's wet, they they kind of create these roots, and but only when they are exposed to light through a hormone, they develop their leaves, and that's the case. Then they know they're out of the soil and they can start photosynthesis, and it would be hindering if they already had their leaves inside the soil. It would need more energy to go up because there's more resistance, and also the leaves could be damaged. And this happens as follows. There's a phytochrome receptor that is activated when light hits the cell, and then the second messengers transport this signal to the nucleus, and later the CA2 plus channels open, and then it is again like the protein kinase is activated to produce like the proteins, etc., that are needed for the leaves to get green. Here's an example of signal transduction. Just a brief question. What would happen, like as you see, it is very complicated. And now, what what you can see is if one receptor or one second messenger is like not working properly anymore. For example, if we deleted this PIF5, then the the function, which is auxin being produced, which causes the elongation um, of plants, would be hindered. But that doesn't mean that it is completely nothing is working anymore, because there are other ways. For example, here it can also have the same effect through PIF4. So um, there are always multiple pathways. Here you see another example of how hormones work. This is the functioning of abscisic acid for stomata closure. As you can see here, it is closed and here it is open, and that's due to abscisic acid. Um, like here, it can't take up CO2, but it also can't lose water. Whereas here, CO2 can enter, and water and oxygen can leave the cells. Here you see like the cell with if there are K plus ions outside it, the water will also move outside, and then it closes. Whereas if K plus is inside the cell, water flows into the cell, and uh, like the stomata opens. Here are examples of different plant hormones that are very important. For example, auxin, which promotes elongation of the shoot, and also um, suppresses the horizontal bud formation in the shoot, and it controls phototropism and gravitropism. There is also cytokinin, which regulates cell division in shoot and roots and counteracts apical dominance, which is like it makes sure that the plant uh, like uh, like width and length are correct, and it also delays leaf aging. Gibberellin is also important as it promotes stem elongation, fruit and seed maturation, as well as germination. Abscisic acid that we saw before inhibits growth and germination, promotes stomatal closure and drought tolerance. As you may know, plants lose water through the stomata, and if they are always open, they also lose a lot of water, which is counterproductive in an arid environment. Um, it also promotes seed and leaf dormancy, so that they, for example, if you have an orange, um, that it makes sure the orange juice contains abscisic acid, therefore, like not a plant doesn't grow out of your orange that you want to eat. Ethylene is also an important plant hormone, which promotes fruit ripening, leaf abscission, and aging processes, and it inhibits elongation growth and promotes horizontal growth and uh, development. Let's talk about ways through which plants can decrease like the effect of uh, arid environments, heat, and loss of water, which is even more important now as we have climate change. There is reduced perspiration in hot and dry areas through crassulacean acid metabolism, or CAM, photosynthesis, um, where only during the night stomata are opened. The downsides of closing stomata is CO2 cannot be taken up, so the plant cannot make photosynthesis, and this is a large problem because plants get all their energy from um, uh, from photosynthesis. Different plants developed CAM independently. This is also called convergent evolution. CAM metabolism is so useful in arid environments that it developed from multiple plants that weren't directly related. So that shows that evolution really tries to find the best mechanisms for to maximize the chances of survival. The problem is that the fixation of CO2 through an enzyme called rubisco is also possible with as oxygen. Oxygen can also bind to rubisco, and that leads to photorespiration, what we'll talk about in a bit. In CAM plants, CO2 is first fixed into malate through the enzyme PEP carboxylase. As you notice, rubisco is used. It is used in all plants, but just the first fixation of carbon, so atmospheric carbon dioxide gas being fixed into an organic molecule that can be later used by the cell or by the plant, is in this case PEP carboxylase, which has a much lower affinity for oxygen, and therefore photorespiration is minimized. The malate is stored in the vacuole of the cell, and the stomata only open during the night. So it is at at different times of the day, the plant cells, the chloroplasts have different function, and not always photosynthesis occurs. And then malate is released from the vacuole back into the cytoplasm. What is the downside of this? Like, what is the problem with closing stomata? You might think not losing water when it is hot is just beneficial, but actually it's not so good because if stomata are closed, you can't take up CO2 and photosynthesis, like you fall down, and it's absolutely not good for the plant. It starves, like all its energy production stops. But if they have their stomata open, they have enough energy and they can produce enough glucose, store it in starch, all good. But then they lose too much water and they kind of fall down this cliff. So they have to find the middle ground um, that minimizes the chances of like dying from thirst and dying from starvation. let's talk a bit about photorespiration. Um, you can see here that ribulose biphosphate plus CO2 react with the enzyme Rubisco and produce a free carbon compound. And the reason why, you may have heard a C-free plants, the term C-free just means the first fixed product of carbon dioxide is a free carbon molecule with free carbon atoms. And the enzyme Rubisco can bind O2 instead of CO2, and then photorespiration occurs. In the light, it consumes O2, which should be produced by photosynthesis. It produces CO2 and consumes energy, ATP, and it also consumes glucose. And this part here is really important. Like, it consumes energy, oh, wait. Like it consumes energy, which is a huge problem because the plant wants to produce energy. It doesn't want to lose energy. And so in hot, dry days with closed stomata, more oxygen is inside the cell, no more carbon dioxide is entering the cell, and the plant dies because it kind of uses its own energy instead of producing it. And in evolution, the concentration of CO2 was much higher in the atmosphere, as you might have known, and the oxygen concentration was much lower, and therefore there wasn't enough oxygen to bind Rubisco. So from an evolutionary perspective, photorespiration wasn't really a problem. There are also, like, scientists aren't really sure if there are benefits to photorespirations. One hypothesis is that that photorespiration decreases the amount of hazardous byproducts of the light reaction of photosynthesis, but this isn't really, like, scientists aren't sure if that's the real reason photorespiration exists. You can see here the what happens in CAM plants during the night. So first of all, you have carbon dioxide, um, which is uh, fixed through PEP carboxylase um, into a into oxal acetate, and that is that is being transformed into malate, and this uses NADPH, which is a reducing agent um, for biochemical reactions. And this malate is stored in the vacuole, so this large organelle or compartment within a plant cell until the next day. And so the fixation of CO2 taken up by open stomata at night makes sure that during the night when it is not hot, the stomata can be open and the plant doesn't die through like losing too much water or photorespiration. But during the day, the malate is again transferred to the cytoplasm, and there carbon dioxide is produced. And carbon dioxide is exactly the thing that can be used to make uh, like make carbohydrates. But the main product is pyruvate, which can be transformed using PEP into a triose, which is sugar with like free carbon atoms, and that can be transformed into carbohydrates like glucose and later amylose and amylopectin, which are the basis for starch. So we'll go over some brief virtual experiment. Um, we want to test how much uh, like malate is present in the morning and in the evening. And as you might know, malate is being produced in the during the night, so the concentration should be higher in the morning than in the evening. And to test that, we have to do this reaction. And as you can see here, NADH plus H plus is produced, and you can actually test this molecule using a uh, uh, UV spectral as as UV spectrometer, and um, but you need to make sure that all malate is transformed. And as you see, it is one-to-one stoichiometry. So one molecule of malate reacts with one molecule of NAD plus with the catalyst MDH, which is an enzyme, and this reacts to oxalacetate and NADH. So one to one to one to one. And to maximize the chance that all L-malate molecules are transformed into oxalacetate, you have to take away the oxalacetate because it is an equilibrium reaction, meaning that if you take away the product, the like the chemical reaction will transfer like the product side will be favored, so ada um, all original substrate molecules are being transformed. And during the experiment, you just continue the reaction of oxalacetate to a different molecule, and now there's always less oxalacetate present than in the beginning, and at one point there is no almalate left. And so you have the amount of NA uh, the amount of NADH which is produced is equal to the amount of almalate that has been produced during the night. And so you can test this molecule in a photospectrometer, and then you know how much almalate has been produced. And we look at the equation with which you can look at it. Um, Here is again what we want to do. Um, uh, you can use the Lambert-Beer Law. And so you use a photospectrometer at a wavelength of 340 nanometers. And through the Lambert-Beer Law, you may know this formula. There are like sometimes different um, terms or like symbols used for it. For example, E could also be an A for absorbance. The epsilon is just a constant which is specific for different molecules. For example, here it's liters per mole and centimeters, and the concentration of the solution inside, like you have the solution with deionized water and the molecules that you pass light through, and you have the concentrations in moles per liter, and D is the path length of the light through the solution in centimeters. So at the end, you get how much is absorbed, and that the more of the solution, the more light the solution absorbs, the more NADH is produced, and that means the amount of NADH produced is equivalent to the amount of malate that has been produced. So the solution is that after darkness, malate concentration is much higher than in the evening, as malate is being produced when stomata are open during the night and used, and the malate is used in the Calvin cycle during the day when light is available and photosynthesis can take place. Let's talk a bit about the drawbacks of CAM photosynthesis. So first of all, CO2 uptake is only happening during the night, meaning that CO2 is like the plant can't really do like there is a limit to how much carbon compounds like glucose it can produce as the uptake of the reactant is limited. CO2 fixation as malate requires energy in the form of the universal energy unit ATP in biology. And energy requirement for energy production is kind of bad because you lose some of the energy that you produce just to be able to produce it. That's similar to how we take up energy through eating, but our digestive tracts also use a lot of energy to process the food. That's kind of similar here with plants. The storage capacity is limited as the malate is being stored in the vacuole, and like even if a lot of malate is produced, at some point a limit is reached, and it can't like it can take up more CO2 and produce more carbon. And therefore the growth rate of CAM plants is reduced. And therefore in environments where a lot of like like in not so hot environments, CAM plants are way less favorable than C3 plants because C3 plants grow much faster, but they are also very prone, like very. Uh, it's really bad if there is a hot period, as photorespiration kind of kills them from inside. But for example, in Europe, where I live, there most of the plants are C3 plants, although obviously there are also CAM and C4 plants. And with climate change, CAM and C4 plants are favored to in comparison to the dominant C3 plants. There are also other adaptations of plants to arid environments. For example, large vacuoles for water and malate storage. You can see the leaves are really big, and here also, like there is so much volume for water and malate storage that it can survive dry periods and also periods with low and available glucose. UV protection, you can see that, but there are actually UV layers on plants because UV light doesn't just damage human skin, and it also affects every animal, not animal, every living being um, as UV radiation damages DNA, and therefore things like cancer and also DNA mutations occur. The leaves have a very small surface. Actually, there are only forms. Sometimes in a cactus, as you want to reduce surface area through which water can be lost. And the photosynthesis does happen in the sprout rather than in the leaves sometimes, or in addition to the leaves. So the leaves can be that small. Here you see a diagram of what happens in C4 plants, which are basically like CAM plants, but they are just not separated by day, but also by where in the cell things happen. Here you see the mesophyll cell and the bundle sheath cell. Here CO2 is fixed also to malate, which is then transferred to the bundle sheath cell uh, where like where it accumulates, and therefore through this accumulation of also of carbon dioxide later, uh, it it the the Rubisco molecule doesn't bind oxygen instead of CO2, so photorespiration is decreased. So PEP carboxylase, the same enzyme as in cam plants, has a much higher CO2 affinity than Rubisco, therefore photorespiration doesn't happen there. And in the Calvin cycle, there is always a high concentration of CO2 for Rubisco, so Rubisco is always surrounded by CO2 and it is not tempted to do photo respiration by binding to oxygen atoms. Here you see the, the the effect of the CO2 concentration and the the netto photosynthetic production and the energy being produced and consumed. As you can see here in C4 plants, which are like CAM plants that don't have photorespiration, they have a sharp increase, like a very steep slope, but they in areas with with higher concentration, it it is much less efficient than C3 plants. And here you see the netto respiration like where the same amount of CO2 is produced as is consumed. And so in dry environments, C4 plants are favorable towards C3 plants, but it is different for environments with enough water supply. Now a question for you. You can write in the chat what you think happens if a C4 plant like corn and a C3 plant like beans are in the same airtight sealed system, like a room or a box without air supply. What happens? You can write in the chat what you think could happen to those plants. Does anyone have a guess? Yeah, exactly. So um, the the corn plant survives longer as it like yeah, exactly. Um, so the thing is also in addition to that, that the corn plant um works even with less CO2, whereas the C3 plant makes photorespiration in that case. So it produces CO2 that can be used by the C4 plant. So if they are together in the same airtight seal container, the corn plant uses the CO2 which is created by the C3 plant transforming amylose and amylopectin into glucose and then glucose into CO2. And through that, the corn plant eats the bean plant. And so it's like a plant that eats just indirectly, um, but not so nice. So yeah. So this was it from the presentation. I hope you could learn something. Um, there will be a, a quiz that I send real quick. Um, Wait a moment. You should now in the chat see the form that you can fill out to receive a certificate. I'll later send it also to the um at in the Discord server, so you can fill it out. But first, let's collect some questions. If you have one, please write it in the chat or unmute yourself to ask questions. So in complete darkness, both plants wouldn't be able to do photosynthesis at all because even the photosynthesis is dependent on light, um, and with without it being present, um, it is like photosynthesis does not occur, so both can't grow, and and it might be a bit misleading. Like sometimes the terminology for the Calvin cycle is the light independent reaction. However, that is honestly not really the case because it takes a few seconds until all the products by the light reaction are like not available anymore by for the Calvin cycle. So um, both the Calvin cycle and the uh, the light dependent reaction are actually light dependent, so nothing would happen in that case.